Good afternoon, good morning. My name is Bert Leufkens. Very welcome on this webinar of the Pediatric Praziquantel Consortium. I think a very important meeting, not only to celebrate pivotal results of the clinical trial program, but also to reflect on the achievements of the consortium so far, but also to reflect on the future of a very important public health problem where the consortium is working now on over a couple of years. My name is Bert Leufkens. I'm the member of the consortium board, also a member of the scientific lead at Lygature. I'm very pleased that I can moderate this session today with you. And we have participants reaching from Japan to Brazil, the US, Europe, Africa, of course. So I think we have a very nice audience here to reflect on this achievement. During this webinar, we will have translations from English to French and French to English. So it's important that we have interpretation services for you available. On the slide displayed now, you will see the instructions how to access the interpretation services. I hope they are very clear to you. When using the services, we recommend that you don't mute the broadcast, the original broadcast, so that you can hear also the original speaker, but that's up to you, but it's most convenient for you to follow the discussions and also to contribute to the discussion. Secondly, I would like to explain how you can interact with us. This is not only sending, we also want to collect your views, your ideas, your reflections. So we will have a Q&A, question and answer session at the end of this webinar. And I'm very pleased also to invite you to contribute to that. And we will also collect questions. So please use the Q&A button at the bottom of your slide, of your screen. And finally, we would also like you know that the webinar will be recorded and will be also available through the consortium website. So these were practical issues, and we have also a warm welcome to those of you joining us and also to everyone that can access some interpretation. So please join us and be also part of our community. So now returning to the topic of this webinar, because I believe we have a wonderful program, not only reflecting on the achievements, but also the long journey that brought us to this space. As mentioned, we are gathering today to celebrate the phase trial data of the arpraziquantel, a potential new treatment option for estimated about 50 million preschool children suffering from schistosomiasis. And to tell more about this journey that will follow, we will have speakers that will reflect on the most recent milestones of the program, but also to reflect on the broader context for the communities in Africa, but also the broader community in global health. So this is the lineup of the speakers. It's a real honor for us that we can start with a video message from Peter Günther, member of the executive board of Merck and also the CEO of healthcare. And Peter's message will be very much on the commitment of Merck, and that is the heart of this whole project. That the commitment of a company like Merck has been so important over all the years and will be also important for the future. And Peter's message will reflect on the commitment, of course, of the company, but also the innovation of the power of innovation, but also to include the communities, the different scientific communities, and also the global health partners in that innovation trajectory. Then we will have Jutta Reinhardt, head of Global Health Institute of Merck, and also chair of the consortium. And Jutta will provide background of the consortium and will also talk us through the whole journey that started almost 10 years ago. And following Jutta's presentation, we will hear more about the clinical details of the achievement so far from Benjamin Delkin, Senior Director of Global Program Health at Merck. And Benjamin will talk us through the most recent results, the clinical results, but also the way we will bring this forward also to the regulatory colleagues to have an important also discussion about the benefit for the children we want to approach. Also, it's important, and that is also at the heart of this whole project, that we always have partnerships in the communities, in the region. So we are very pleased also that we have Professor N'Goran from Côte d'Ivoire on the floor. He will also reflect, he was the PI of part of the clinical trial program, and he will reflect on how the region, but also how the community in his environment also have contributed not only to the clinical trial data itself, but also how the results will also used for the future treatment scenarios, but also how to reflect on new possibilities for collaboration. The next speaker we will introduce is Peter Steinman, and a very important colleague from Swiss TPH. As you well know, this is a partner very much involved in global health for many years. And Peter is co-lead of the ADAPT program, and the ADAPT program is very much also focusing, okay, at a certain moment there will be a formulation for pediatric application, but how can we implement it, adopt it, facilitate also the adoption in the regions. And Peter will reflect on that, in particular also how the 5 years ADOPT program will facilitate in terms of multidisciplinary involvement, also efforts that can also bring key stakeholders to the floor. And of course, also at the end of the day, ensure acceptance and equitably access to the treatment. Because if we don't reach these 50 million preschool children, then I think we don't have a successful program. So this will be key of the whole consortium. And finally, we are very honored, but also very pleased that we have Dr. Magange, CEO for the consortium funding partner, EDCTP, because this funding is not only essential for this whole program, but also a key partner also in future thinking, but also to put this whole project in the context of learning. Because this is a single treatment for a single disease for this community. But the broader perspective, of course, of this consortium is how can we learn from all these, steps also to bring new future treatments also to the global health program. So we are very pleased, Michael, that you are participating here, and we are looking forward also to your contribution. As mentioned, time will be also made for questions and answers. So we are very happy also if you use the chat box as much as possible, so that we also collect these questions and so that we have also an interactive way of bringing this forward. So thank you again for all this, but thank you also for being with us. I see here at the calculator that we have more than 110 participants now. That's excellent. If you look also at at the geographic distribution and also at the distribution in terms of organizations. It's public, it's private, it's global health, it's science, it's also patients and also the clinical community. So we are very pleased with this very broad audience. So a quick reminder that you can have access to interpretation service again. So use the button on your screen. And again, it's important also to have that reflection on the screen. So having said that, I'm very pleased to introduce the first speaker. He will speak to us through video, please.
Good morning, good afternoon, good evening. Thank you and a warm welcome to all of you for being here with us today. I'm honored and proud to be able to celebrate an important development in the fight against schistosomiasis together with our partners of the Pediatric Praziquantel Consortium, namely the positive phase 3 results for arpraziquantel to treat very young children. Schistosomiasis is the second most devastating tropical disease after malaria and critical due to its economic impact. It can lead to impaired growth, learning disabilities, and chronic inflammation that can be fatal in the most serious cases. Today, we provide a proven treatment mainly for school-aged children. However, the treatment of over 50 million preschool-aged children has remained a significant challenge, mainly due to the lack of a pediatric medication. The positive phase 3 results for arpraziquantel now provide a potential solution to this critical unmet medical need and pave the way for submitting the registration dossier in 2022. For over 14 years, Merck has engaged in the fight against schistosomiasis and more recently committed to the elimination of this disease as a public health burden. We have adopted an integrated approach that goes beyond donation and invests into health education, sanitation, and hygiene initiatives, as well as R&D for new drugs and diagnostics. At the end of the day, we harness the power of innovation as a leading science and technology company and build equity and access to health to leave no one behind. Together with our consortium partners, including those from endemic countries, we have pooled our creative minds and expertise and leveraged our shared purpose to positively impact people's lives. In addition, in this private-public partnership, we have established a powerful model for developing therapies for vulnerable populations. These therapies have the potential to break the cycle of disease and poverty and address socioeconomic challenges by enabling sustainable development of communities. A critical factor for the success of the model has been the full integration of the countries and their expertise into the design of the program. This collaborative effort has been a great journey for all of us in the consortium. For this, I would like to congratulate the team, all consortium partners and collaborating experts, including the WHO, for their exceptional work in reaching this milestone. I wish to also thank the Bill and Melinda Gates Foundation for their contribution and EDCTP and GHIT Fund, who have gone far beyond simply providing financial resources, but accompanied us to this wondrous achievement. The success of the Pediatric Praziquantel program is certainly a concrete example of the strength of our common approach to sustainable and innovative collaboration in global health. With the ongoing efforts of the consortium and the continued consolidation of local and international partnerships, I am confident that the elimination of schistosomiasis is a goal that can be reached.
Well, thank you, Peter. I'm sure that words of appreciation will be feedback to you, and I think we feel really honored that you were able, through video, but you were very close to us, to express your views on this important project, but also to make links to all the partners, WHO, the communities, the regions, etc. So I think that was wonderful. So to continue with the program, I'm very pleased to invite you to our next speaker, Jutta Reinhardt. Jutta already introduced her briefly in the introduction, has a lot of titles in terms of the lead of the Global Health Institute, but also she drives a lot of other initiatives. But to me, and I learned her very much over the years as member of the board, and she was chairing the board, in particular when we had the consortium meeting in Kenya a couple of years ago. She is a true champion of global health, and it's so particularly important that companies like Merck has these wonderful people on board, because she can bridge from a corporate level to the global health perspective. And Jutta, I'm very pleased that you will reflect with us on this long journey, because actually you were much more longer involved than myself. So I'm very pleased that you can bring us back to where it started and why it started.
Thank you. Thank you, Bert, and thank you so much for this kind introduction. I feel really a bit embarrassed, I have to say. But I'm happy to share with you the history and the journey. Is this in presentation mode? Just a second. Yes, I think now we are in presentation. Yes, super, thank you. You heard already from our Merck CEO of healthcare, Peter Günther, about schistosomiasis. I think the community that is listening today, I don't need to explain much about this disease. It's mainly in Africa. Over 90% of patients live in sub-Saharan Africa. But you will see from our partners, Brazil is an important partner. Brazil is also still a country that is affected by schistosomiasis and that really would like to eliminate the disease. We heard about how it's caused and it's spread through water. Let me just say a few words about the consequences of this disease, because over the years, when I was working, and I'm still working on this disease, I got really to know more about the impact of long-term consequences. And, you know, I heard from a colleague in Mali about the liver fibrosis in 4-year-old children. I mean, how is this possible? How can we accept such a severe disease, a consequence in so young children? We hear about girls with genital schistosomiasis. Bladder cancer is a consequence. So really dramatic consequences of a disease that goes nearly unknown, unnoticed, because it's the disease of neglected people. We came together, yes, 10 years ago, we came together and triggered, I tried to think about what was really the trigger. So there were really a few people outside, one to mention, and apologies if I cannot mention everybody, but really one person who influenced me a lot was Russell Stoddard, who wrote already a paper about the neglected population of these young children. I try to get to the next slide, but it's not working. Thank you. So this neglected population of very young children, younger than 6 years of age, we call them preschool-aged children, they are not treated. And this really causes an ethical concern. We had quite some discussions whether they are infected or not, but there was at least anecdotal evidence that these preschool-aged children can also be infected. And you heard from me before that we had liver fibrosis, genital schisto. People have seen it. But still, there was quite a discussion in the beginning, quite some hurdles to overcome, whether this is a high medical need, whether there is really a high prevalence. We did not give up. We found partners. I think we started with Astellas, Swiss TPH, and Lygature at that time. And then Pharmangenius joined shortly after that. We started a consortium, a public-private partnership to address this unmet medical need. And we formed what we call today the Pediatric Praziquantel Consortium. So what is this consortium about? We really have the vision to reduce the burden of this disease, to address the unmet need in these preschool-aged children. This would include infants and toddlers. This is really young. And you will see later, this is really a challenge. So our mission is to develop, register, and provide access to a suitable pediatric treatment for schistosomiasis in this age group. And I'm emphasizing to provide access. So this is where a second stream of our consortium comes in now. And we had webinars for this a while ago, the project called Adopt. But today we talk about the results of the past 9 years on the development part. So before I enter into what we have achieved, let me just give you a bit of background on this consortium. To my knowledge, it's the first international public-private partnership on schistosomiasis. We established it in July 2012 as a non-profit consortium. We have partners that joined us during the life of the consortium. We had partners that joined for a while because they had a certain task to fulfill and then they left, and they still, of course, follow what we are doing. But it was really it's an open consortium. And depending on the need, and we started really on the preclinical when we started, depending on the need, partners came in or also left. We have a solid governance structure. We have a consortium board, as you have heard from Bert. All the partners are represented at this board. And then we have a development team. That's our focus of today. And we have an access team and also several sub-teams. We are supported by international experts and funders, and we are very, very proud that also WHO is instrumental and is an observer. You see here all our consortium collaborators: Merck, Astellas, Swiss TPH, Lygature, Pharmangenios, SCI, Khemri, the University Félix Houphouët-Boigny in Côte d'Ivoire, Klinikum Rechts der Isar, that's the Technical University in Munich, the Ministries of Health from Côte d'Ivoire, Kenya, and Uganda, AIHD in Kenya, and Makerere University. So what a development of this consortium over the years. When we started in 2012 with 4 partners, fighting a bit to justify the existence of such a consortium, the need that needs to be addressed, and now having these many partners so much committed. So our goals that we wanted to implement. It says in our mission to develop and launch an innovative pediatric medication to treat preschool-aged children that are suffering from this disease. So what we had, and that I think is a key recipe also for other partnerships, we conducted expert meetings. We asked really the experts from the field. We discussed target product profile with key opinion leaders, with WHO, with the countries. We had regulatory path meetings with experts early on. And looking into the future, we also discussed the collaborative procurement model. How do we ensure affordability? Because we know the patient, the young child, cannot pay for this medication. So we were looking for sustainable ways to procure praziquantel pediatric formulation for this patient group. And what are these sustainable mechanisms? So we integrated the access stream quite early, I would say, already 3 years ahead of launch. So again, expert meetings, but it also led to the development of a whole new work stream, a whole new project within the consortium. We achieved local manufacturing. So we work with African manufacturers, so they will manufacture the product in the future. And as I said, it is really important to say treatment remains free for patients. And not to talk about sustainability goals, I think you understand easily how this project supports SDGs like number 3, number 9, and especially 17, where it says partnerships to achieve these goals. I think our partnership is a wonderful proof for this. So what do we have in hands? We have an oral dispersible tablet developed. It is called arpraziquantel. For the chemists among you, you know that praziquantel is a racemic mixture of 2 enantiomers, but only one is active. And there might be colleagues in the audience who worked on the enantiomer and who have shown that it's also less bitter. So we became quite interested in this enantiomer, and we had quite some assumptions and expectations with this enantiomer. It's smaller. Our tablets today are easier to use. The palatability is improved. We can dose it much more precisely. It's, as it says, orally dispersible. It can be taken with or without water. And the bitterness is reduced. It's not completely gone, but it definitely has a difference. If you know how praziquantel tastes, you will immediately agree with me. We had to ensure stability in hot and humid conditions of tropical regions, very clearly. And we really aim to transfer the production to local manufacturers, and we have to ensure that this can be done at a large scale. So we could not jump immediately into clinical development. We still had some work to do on the preclinical side, despite the fact that praziquantel is known since more than 40 years. It's on the market since 1980, I think. It's tested in clinical studies. But I would say not a lot, because it's a neglected disease. Not a lot has been invested in further research. So we still had to do some homework, especially working with the enantiomer. But then we were able to enter clinical development, and interesting discussions in Europe about the taste. Is it bitter? Is it not bitter? And then we said, why are we discussing this? We have to ask the relevant population. So we need a taste study in African children. And that's where we started in 2015. Very interesting taste study in school children, because the younger ones are, of course, much too difficult to target and to express their reaction to the taste. But here with the school children, we had quite a success. We were quite pleased how acceptable the ODTs were. And we continued with phase 1 bioavailability studies in South Africa. This is in, of course, healthy adults, healthy volunteers. And then it became really exciting when we took our product into phase 2 for PK/PD dose finding in Côte d'Ivoire. Côte d'Ivoire, we had, as the species schistosomiasis, we had children from 3 months to 6 years. This is, for me, most most astonishing, and it was a huge effort to find all these children in this age group. Many children were screened, and I think our colleagues in Côte d'Ivoire did a great job here. So with this PK/PD dose finding, we were able to go into the confirmatory trial in phase 3, and this was done in Kenya, in Côte d'Ivoire. Kenya also has hematobium, so we also included children, young children infected with hematobium. This study, of course, we were facing challenges with COVID, we had clinical holds, but we were able to finish this study just recently, and you will hear about the results in more detail from Benjamin right after my presentation. And this takes always a little bit. So just to conclude, what is important? I think with our vision and mission, I can say we are following what we said in the beginning. Whatever we do, we look at it really from a local capacity aspect, capacity strengthening aspect. So when we do clinical development, I would say we were really able to strengthen infrastructure for clinical sites. We did trainings with local healthcare professionals. We could maintain the skills and the teams during COVID and clinical hold. I think these are very important aspects that nobody had foreseen. On the manufacturing side, I mentioned it several times. We have an agreement now in place with Universal in Nairobi for large-scale production, and a lot of knowledge that started in Japan, in Germany, had been transferred to Brazil, is now going to be transferred to Kenya. I think the colleagues here on the CMC side and the manufacturing side are doing an incredible job. And this is truly global. I tried to get to my last slide, and I wanted to thank you for your attention. Over the years, we have also developed a very nice website. I think you hear also regularly from us. We have a communication team that is very active. I want to thank all the partners, but also all the colleagues that are involved and that were involved in the past for a period of time. They all believed in this project, and only all together we could come up with these results and with such a success. Thank you very much. And I hand over to Benjamin. Thank you.
Thank you, Jutta, and thank you also for this very in-depth reflection on the long journey. I really appreciate your comment. What do we have in hand? Because this was not only a talk shop, this was also very concrete, and particularly also the very interesting marriage between the CMC part and the clinical development. So, and having said that, I'm very pleased that we can continue with the program. As already said, Benjamin Delkin, long history and long experience in the industry, will reflect on the results of the pivotal trial already mentioned by Jutta, but now we go into the details. Benjamin, the floor is yours.
Yeah, thank you, Bert. I'm today really very pleased that I can share with you positive results of our phase 3 trial. You see here in the scheme in the white part, you see that the trial was conducted, as Jutta mentioned, in Côte d'Ivoire. Here we enrolled schistosoma manzoni infected children, and in Kenya, where we enrolled both schistosoma manzoni and hematobium infected children. So we agreed the trial design with the European Medicines Agency before the study start. You see that in the scheme on the left side, we had the trial separated in the preschool-aged children that were either infected with schistosoma manzoni, shown in blue here, or with schistosoma hematobium shown in red. And we structured the trial in different groups according to the age of the children. All children received a single dose treatment of either arpraziquantel, and this was at a dose level of 50 mg per kg, or biltricide, which we included as a reference just to compare the data to. And this was used at a dose level of 40 mg per kg, the standard dose that is at the moment being used in the control programs. When you look at the different cohorts here in this blue scheme, you see cohort 1. Yeah, here we had 4 to 6 year old children enrolled, and they were randomized in a 2-to-1 ratio to either receive arpraziquantel or biltricide. So these were the only randomized cohorts we had in the trial. Then we had in cohort 2, 2 to 3 year old kids. And in cohort 3, we had the very young kids, 3 months to 24 months of age. And because this cohort was hard to enroll, we pooled the results together with the very young kids that we enrolled in our phase 2 trial. When you look at the light red boxes here below, schistosoma hematobium infected children, here we had one age group from 3 months to 6 years old children. And we also initially started with a dose level of 50 mg per kg. So again, we had no experience with arpraziquantel in schistosoma manzoni, in schistosoma hematobium in the phase 2 trial. That's why we had the dose finding element here in the schistosoma hematobium species implemented into the trial design of the phase 3 trial. And we saw after the first 30 patients where we did a preliminary readout that we had to increase the dose, and we did this to 60 mg per kg. So I will show you the results for both those levels later. We also included a second time point. So all the readouts we did in the trial for cure rates, for example, we assessed at week 3. And here for the hematobium infected children in cohort 4B, we also included week 5 assessment. Here you see the key endpoints that we used in the trial. Primary endpoint was the cure rate at week 3 in schistosoma manzoni infected children in cohort 1. And here we had a pre-specified threshold for the cure rate. And this was true for the lower bound of the 95% confidence interval. And this had to exceed 70%. So this was, so to say, our expectation and also our benchmark we included here. Secondary endpoints included cure rate at week 3 and week 5 in all remaining cohorts. And here we used the same threshold for the cure rates with the lower bound of the 95% confidence interval to exceed 70%. Reduction of X from baseline was another important secondary endpoint and was assessed in all cohorts at week 3 and in schistosoma hematobium infected children with a higher dose level also at week 5. We also assessed, of course, safety, very important, and palatability. Here you see the selection criteria of our patients that we enrolled in the trial. So they had to have an age from 3 months to 6 years, and then they were distributed over the different cohorts by age group. They had to have single infection with either schistosoma manzoni or hematobium. The body weight had to exceed 5 kilograms. And then, as usual in pediatric trials, we obtained for all patients parental consent. In the trial, we excluded patients with mixed infections, medical conditions that would jeopardize patient safety or study readouts. We also excluded patients with a medical history of seizures, cysticercosis, and debilitating illness, like, for example, tuberculosis and malnutrition. so you may realize that all these exclusion criteria basically were taken from the Praziquantel label. We also excluded concomitant treatment that would disturb the metabolism of Praziquantel. And treatment for malaria 2 weeks prior to screening was also excluded. Here see the disposition of the patients that we included into the trial. So we had to screen quite a lot of patients to find the right patients. Approximately 2,700 patients were pre-screened. And we screened 326. And we enrolled, randomized, and treated 288. And this was defined as the safety population. Then we excluded 8 further subjects from the safety set due to the use of malaria treatments during the trial. And this defined the MITT population, so the patient population where we assessed the cure rates in. You see the demographic of the MITT population. So please note that in cohorts 1A and 1B, we had, according to the study protocol, a predefined minimum of moderate and heavy infected patients that we enrolled. And this minimum was 40%. And you see that we achieved approximately this 40% in both cohorts. Whereas in the other cohorts, infection severity was lower, except for cohort 2, where you see we had infection intensity of almost 60%. So in conclusion, we think that this infection severity that we saw is consistent with a real-life setting, except for cohort 1, of course, where we the proportion of moderate and heavy infection was pre-specified. Here you see the cure rates as primary endpoint. Please look at these first 4 green bars first, schistosoma manzoni infected children. You see here that we achieved cure rates close to or higher than 90% with these 3 arpraziquantel cohorts. Here you see the reference group, Viltrucid. Here we reached 81%. And then I've also included here as the dotted line, the efficacy threshold that we wanted to reach, 70%, with the lower bounds of the 95% confidence interval. So these lower bars here that you see around the point estimates. So you see that for arpraziquantel, we exceeded this 70% threshold in all those groups, which made the study from the cure rate perspective a success. You also see that Viltrucid is slightly crossing the 70% threshold. However, please note that here in the randomized cohorts, cohort 1A and 1B, we had, due to the randomization, we had a lower sample size here in the Viltrucid cohort 1B, and consequently also a wider confidence interval around the point estimate. So probably if we would have enrolled more patients in the Viltrucid group, then probably we would also have exceeded the 70% threshold for the confidence interval of Viltrucid. When you look at the schistosoma hematobium infected children, here represented with the blue bars, you see why we increased the dose. You see that for the 50 mg per kg dose group, we obtained approximately 60% as a point estimate with the first 30 patients. And that's why we increased the dose to 60 mg per kg. And you see here with the higher dose level, we obtained better efficacy, 86%. And this was a normal week 3 time point. And then please remember that we also included for schistosoma hematobium, the higher dose level, another assessment time point at week 5. And this is shown here. Yeah, you see 95%. So the efficacy in terms of cure rates further increased, although the confidence intervals are, of course, overlapping. So probably it makes sense to look, this is our learning, to look at different time points, especially for schistosoma hematobium, also at later time points to really understand the efficacy. Here you see the pooled cohort of the very young kids in, from our phase 3 trial and from the phase 2 trial. We obtained a point estimate of 95% for the cure rate, very consistent with the very young kids of the phase 3 trial, and also very consistent with the very young kids in our phase 2 trial. So bottom line is the pre-specified threshold for cure rates was met in all cohorts and across species that we assessed. For schistosoma hematobium, this is only true for the higher dose level of 60 mg per kg. You see the reduction of X from baseline. A group-based geometric mean is represented here. And you see that we obtained 99% in all cohorts. This predicts disease control and morbidity control, and also is exceeding the, let's say, WHO defined value of 90% for reduction of X. Safety results very important in terms of TAEs. You see here that the subjects with any TAEs or related TAEs, and you see here that the numbers were generally higher in the schistosoma manzoni cohorts compared to schistosoma hematobium. But you also see that the results between the randomized cohorts 1A and 1B are very similar, so very comparable results between arpraziquantel and biltricid. We did not see any serious related TAEs, no TAEs leading to discontinuation or death. So bottom line here is we think that in terms of safety, we have similar results between arpraziquantel and biltricid here in this trial. You see the most commonly IMP-related TAEs, and they were in the system organ class of gastrointestinal disorders. Again, you see higher numbers in the schistosoma manzoni infected children, cohorts 1A to 3, and lower numbers in the schistosoma hematobium infected children. But also here, we had very comparable results in the randomized cohorts 1A and 1B, so very comparable between arpraziquantel and biltricid. You see here in cohort 2, we had higher numbers in the system organ class. And this is probably due to the higher infection intensity we had in this cohort 2, where we had 60% of patients with moderate or high infection compared to 40% or lower in the other cohorts. We had some cases of somnolence, a few cases, but also here very similar in the randomized cohorts when we compare arpraziquantel to biltricid here. So in conclusion, gastrointestinal disorders were most frequent TAEs that were reported for both arpraziquantel and biltricid, and also what we would have expected from the experience with praziquantel. You see the palatability score. You see that, please note that the palatability was assessed by parents, and here only in cohorts 1 and 4, and only in children with an age of 5 to 6 years old. So we have the older kids where we assess the palatability as in the palatability study. And you see here that with the new tablets, palatability was improved compared to the control cohort 1B here. This brings me to the key conclusions of the trial. Efficacy and safety data shows a favorable profile. And here, especially when we look at our primary endpoint cure rate, but not when we look at the egg reduction rate, then this is only true for the 60 mg per kg for the higher dose group for schistosoma hematobium. The study met its primary endpoint, which means that the lower bounds of the 95% confidence intervals exceeded the 70% threshold that was pre-specified. The egg reduction rate was very high, with 99% in all dose groups, in all cohorts, but also with both dose levels that we used in schistosoma hematobium. We did not identify any new risks or safety concerns in the trial. And in terms of safety, both dose levels, 50 mg per kg and 60 mg per kg for arpraziquantel demonstrated favorable safety, tolerability, and improved palatability among preschool-aged children. In the end, I really would like to thank all consortium partners who made this trial possible. Of course, the clinical trial staff, the investigators, and special thanks, of course, to the children and their parents. Another special thanks also to the funders here, EDCTP, the GHIT Fund, and the Bill and Melinda Gates Foundation. Thank you very much, and back to Bert, I guess, right?
Yes, thank you, Benjamin. Very interesting, very relevant data, and a lot of data, but overall was a very strong message. The product works for the target population. I think very nice, and I saw already in the chat box that we have a number of questions, details on your presentation, but we will come back to that later on. So again, thank you. We will continue with the program, and I'm very pleased, as I said before, that Professor N'Goran is here present. Professor N'Goran is the local PI for the consortium, the clinical trial, and together, as Jutta already showed, together with colleagues from Kenya, a key leader in the field, and also a key figure also in this trial. So, Professor N'Goran, we are very pleased that you will share with us your experiences in this clinical trial program. Thank you.
Thank you. Good evening, ladies and gentlemen. Dear seminarians and distinguished panelists, it is a great honor and pleasure for me to take the floor and share with you on behalf of the investigation team the exciting experience that we've been able to experiment through this clinical trial. After a brief introduction, we will therefore present the challenges, the acquired experience, and the lessons we've learned before we reach a brief conclusion. As it was mentioned in the previous presentations, the Ivory Coast was honored to engage in phase 2 of the clinical trial, aiming at developing the formulation and the efficient dose of the candidates to treat preschool children. It is an experience that lasted from 2016 through 2021. The major challenge of such an experiment was to lead the clinical trial in compliance with the good clinical practices defined by the International Conference of Harmonization of Technical Requirement for the Registration of Drugs for Human Use. We therefore were to follow the so-called E6 guidelines. Now, to lead such a trial with such a quality standard in a developing country required to take into account a series of hindrances to be overcome. And therefore, I will share the Ivory Coast experience with this background. Naturally, such an experiment was published in 2018. As you know, the Ivory Coast is a French-speaking country. Unfortunately, the language of science and also sometimes the language of clinical trials is English, usually. Second concern, the Ivory Coast has spent this last decade among a social political turmoil that has questioned a series of progresses and that has led communities and populations to become suspicious. Concerning the fight against schistosomiasis, since 2013, thanks to the support of the Schistosomiasis Control Initiative and of the British government, populations suffering from schistosomiasis are administered a preventive chemotherapeutic treatment. Here we should point out that Merck also provides drugs for the treatment of school-aged children in the Ivory Coast. The trial took place in Man and its surroundings in the Tongpi region and in the neighboring regions, around 600 kilometers away from Abidjan, where the Félix Houphouët-Boigny's university headquarters are. And the university is actually where the coordination of the teams took place. The area of Man, in its extension and given its main features, may be considered as a representative of the endemic sites in the real world that need to be visited if we want to treat affected populations. Now, let's look at the challenges that we had to face. The first one was accessibility of the sites. We have mentioned that this is a quite extent area. So for the phase 3, we were in the Gemon and the Tonki region. For phase 2, we even added the Cavalli region. And in phase 3, for the 100 participants, we had to visit 29 villages between February 2020 through May 2021. So you see in the picture how difficult it was to access the participants in these sites and to transport them to the health center. We may point out 3 main challenges that we had to face. The first one was, as was mentioned, capacity building. By capacity building, I mean infrastructures, then equipment, and also the qualification and training of the investigation and operation staff. The second main challenge was to engage communities in the study. And finally, the third main challenge was to comply with ethical and regulatory approvals for such clinical trials. Concerning the capacity building, to carry out such clinical trial, we had to actually build a facility dedicated to the participants in the project because, first of all, we had to set them aside from the usual hospitalization rooms. So we had to set them aside from the rest of the patients of the hospitals. And also, we built this facility because there weren't enough beds to be dedicated to the trial. Later on, offices were also set up to facilitate and improve the development of the trial. On the other hand, we had to equip the labs to be able to carry out all required biomedical tests. Then came training of human resources. Naturally, for several of the members of the investigation team, it was their first experience in a clinical trial that complied with the standards of the International Conference of Harmonization. Therefore, we needed to carry out intensive training and capacity building sessions so that the members of the team could comply with the quality requirements. Concerning the community engagement, it wasn't easy given the general background of turmoil and socio-political conflict. To overcome that issue, we had to implement a communication policy that involved all administrative authorities, starting with the prefect of the region, who conveyed the information to the sub-prefects, who then informed the chiefs of village. And only then did we meet with traditional chiefs and community leaders to share information and discuss with them to get them to engage. Obviously, we always awaited their agreement before starting any treatment. Also, there were areas that spoke several languages. In that case, we needed interpreters, and often from dialects, so that the populations could understand us. So these were the requirements of the project. And such an approach, we think, was an important element in the overall success of the recruitment we were able to carry out. Other challenges had to do with the population's informed consent due to illiteracy. Sometimes we missed identification documents of children, and this prevented us from recruiting a good deal of children. And also there was a lack of independent witnesses to co-sign the informed consent forms, and then there were rumors on the destination of blood samples, etc. And the last great challenge was therefore, the regulatory compliance. In our experience, actually, the implementation of the clinical trial was a great opportunity for the Ministry of Health because it helped it clarify the regulatory submission procedures to obtain authorizations for clinical trials. And to succeed, we had to organize several meetings between sponsors, investigators, and the authorities of the Ministry of Health to clarify all this. Because when we started, there were different interpretations of the regulatory requirements depending on the body in charge of the clinical trials. But in the end, in 2018, an external assessment took place to clarify and define the procedure for the authorization of clinical trials. And this led to the development of an Ivory Coast pharmaceutical regulatory authority in 2018. And naturally, this newly created body is in charge of approvals and authorizations. Of course, this process shows that sponsors and stakeholders need to be patient and use dialogue in order to resolve any issue that may occur throughout the trial period. What to conclude? We may conclude with the following statement. It is essential to carry out clinical trials in developing countries to overcome the diseases that affect the populations who live in extreme poverty and sometimes on the verge of development. Obviously, our experience here shows that in order to reach standards, we need to be able to adapt international guidelines to the developing country's background. Therefore, we think that this experience may be used as a case study that shows how a public and private partnership may lead to quality clinical trials on diseases that affect populations in poor or developing areas. Such trials lead to improved skills and capacities among domestic institutions by looking for pragmatic solutions to issues that arise and by improving the general environment in terms of regulatory requirements for clinical trials. This is, ladies and gentlemen, what we had to say. And before ending, I wish to sincerely thank all the populations that took part in this experience, parents, children, investigation team, as well as all partners, thanks to whom this study was carried out. And I would also like to add, I wish all these skills end up being developed in developing countries so that research and diseases that affect developing countries or research on diseases that affect developing countries is carried out in compliance with international regulatory requirements. Thank you very much.
I feel very impressed, but also deeply humble when I see as a scientist, but also as a former regulator, that when we have all these tables and data on the table, how much effort and the local communities have been invested to bring the data at the end of the day to the table where decisions have to be made. So congratulations, and also deeply impressed by your contribution. I'm sure we will come back to that later on in the discussion, but we will continue now with Peter Steinman. He, as I said before, he's the co-lead of the consortium and particularly involved in the access adopt part of the project. So Peter, the floor is yours.
Thank you very much for giving me the floor here and the space to talk about the ADOPT program. The ADOPT program and the access team activities. Why do we have these access team activities? Has been introduced already by Jutta at the very beginning, since the consortium mission is not only to develop and register the new drug, but also to provide access to it. And this, of course, are very different activities. And Also, therefore, we have divided the activities into two teams, under two teams. Development team, that's what you have heard until now, the trials, and this will be followed up with the registration of the new drug. And then the access, which includes the technology transfer, the actual delivery, methodology, the funding to ensure sustainable access to the new drug over time once it has entered into routine healthcare provision. Now this, yes, here. It is very important that access considerations start to be taken before the drug is available to ensure that the patients in need, in this case, the preschool-aged children, have access to the new drug as early as possible after drug licensure. And therefore, already 2019, 2020, we started to develop a program. This is called the ADOPT program, which would combine all these activities necessary to ensure access. We developed a proposal and we received funding for a 5-year program from EDCTP and GHIT, with activities starting already earlier this year, 2021. And we feel, of course, vindicated by this early start, by the positive results of the drug trials, which have been presented just now by Benjamin, so that we see this was really a timely investment into future activities. The ideas guiding the ADOPT program are summarized here. So the goal of the program is to understand the perception of the pediatric schistosomiasis of the disease in the communities, but also its treatment, so praziquantel-based treatment of schistosomiasis. And then based on this understanding of local perception, to define social mobilization strategies which can support the distribution of pediatric praziquantel in these communities. In parallel, we have to identify platforms and approaches to deliver the drug to the preschool-aged children. Remember that the traditional way to deliver the traditional way to deliver schistosomiasis treatment is through school distribution, mass drug administration at school level. Now we talk about preschool-aged children, so we don't find them in the place where the traditional schistosomiasis control programs work in the schools. So we have to identify new platforms, new channels to reach this population, these preschool-aged children. We defined 2 steps for the implementation program. First, as mentioned, the social mobilization communication strategy, which has to be tailored each time to the national health system and the context. And then a staged implementation process, which would lead in the end to the most appropriate platforms and approaches. Staged in the sense that we start with, at small scale, comparing different approaches and then selecting the most performant one and scaling it up to larger population groups. And then in parallel, we have the identification of the funders and stakeholders, which in the long term can support the routine implementation of schistosomiasis control in this preschool-aged children population. Here, the ADOPT program, the aim is really to prepare the introduction of the levopraziquantel 150 milligram tablet into the schistosome endemic communities. And this has been defined as a multidisciplinary effort, which requires the engagement of all key partners and stakeholders involved in this process. First, starting with a set of feasibility studies, but also cost effectiveness to ensure then the acceptance at community level, but also at the health system level, ensure equitable access to treatment for the population, and describe the validated approaches which can then be taken up by national programs, taken up into policy change, and rolled out in the countries which we target now, but also the other endemic countries, which one by one will certainly become interested in adopting the new drug. We have defined this as a toolbox for adoption, so a set of very concrete guidelines which will facilitate the uptake in countries, even if they don't have the experience of the clinical trials and also the experience of the ADOPT program. Then we have the technology transfer to the manufacturer in Kenya. Again, Jutta has mentioned that, so that we achieve local manufacturing of the drug and the resource mobilization to support the scale up beyond the study as we have described here in the ADOPT program. The objectives, we have defined 5 of them, 5 key objectives. First, to identify potential facilitators and barriers to the implementation of the praziquantel delivery and inform thereby the appropriate social mobilization, communication, and training strategy. Then, as second key objective, to assess an existing and then select the optimal access platform, the delivery approach within these platforms for the new drug. Number 3, to quantify drug and funding needs for integrating the pediatric praziquantel into routine schistosomiasis control. This is extremely important so that we know what we talk about in terms of manufacturing needs, in terms of funding needs, which need to be covered from sources in the countries or from international sources. Then to document the safety of levopraziquantel administration under the programmatic conditions. Of course, we have safety data from the trials, but in the real life situations, depending on the platform, the drug may be delivered together in parallel with other pediatric health products, and we need to be sure that there are no interferences here. And then last but not least, number 5, to inform at national level, but also at international level, the policy change which is necessary to adopt this new program, give recommendations and backing at highest level to the introduction. Here, I have summarized on one slide the kind of an overview of the ADOPT program. So it's an implementation research program. It's not designed as a trial. It's really very close to reality, as close as possible, bringing together all the relevant stakeholders from the pharmaceutical company level, academic institutions, but then also NGOs, government from endemic countries, which all need to work together to achieve the aims and objectives of the ADOPT program. The program is divided into 2 stages or 2 phases. One phase, pre-licensure, that's where we are now. Everything what can happen before licensure of the drug, preparatory activities, and then post-licensure, once we have the drug available for use in the population, the work in the field with the delivery and the selection, the final selection of these platforms and the toolbox development. So we have a staged approach from identifying, piloting, evaluating, maybe adapting, going to upscaling and evaluating again, so that we always learn from one stage to the next and can ultimately come up with the optimal solution. 5 years duration, I have mentioned before, and we have this program running across 3 countries. You recognize Kenya and Côte d'Ivoire, which have been involved in the phase 3 trials already. And then Uganda has joined also to have more variety in terms of health systems, in terms of local settings. The outputs and outcomes include, of course, the recommendations to support the introduction into routine practice in community settings, and then the mentioned toolkit, which will be available not only for these countries, but for all the endemic countries to pick up as quickly as possible the learnings. Here, a graphical overview over the different work packages under the ADOPT program. Of note, results from one work package always feed not only in subsequent stages of the same work package, but also into stages, activities of other work packages. So they are all interlinked. Just as an example, work package 1, the social science studies, directly inform advocacy and social mobilization activities in work package 2. These again are of course an integral parts of the access strategy pilots and then district level distribution studies in the countries, and monitoring evaluation going with it. Where do we stand with this whole program until now? The status is that we have all the partner institutions set up. We have all the teams in the partner institutions working. We were, despite all the travel restrictions, able to have planning workshops in each participating country. This was very important to have this direct, immediate contact with representatives, not only of the consortium members, which we know already from previous work, but also with other partners in the country, stakeholders, which are relevant, but are not members of the consortium. Then we have the social science study protocols developed. We have submitted them for ethical clearance in the countries and are waiting for the outcomes of these applications. And along with this, we are of course advancing with the general preparations for the developing all the tools, the platform selection, development of the M&D tools, data management, and so on. And this brings me already to the end of my presentation. I'm happy to answer questions later. Thank you very much for your attention.
Thank you, Peter. Very insightful, and also this very very important switch also to the social sciences and also the mobilization of the communities to make this a success. So excellent. So we now have opportunity to interact and also to have discussion with the panel. I saw already a couple of questions through the Q&A. I saw also some other questions fueling in. So please, if you have questions related to the presentations, related to the project or the consortium as a total, use the Q&A so that we can have as much input from you as possible. I would like to start with a more general question, and maybe it's related to, I think it's related to the presentation of both Jutta and I think also Peter, but also Professor N'Goran mentioned that the importance of early dialogue, of early interaction, and early is a very fertile concept, because often you are too late or too early, etc. So, but this early dialogue, particular, and I think both of you mentioned that, to build trust. Could you reflect a little bit more on that, how critical that is, particular for this kind of project, but maybe also more special also for this project we are talking about? And maybe you could start with that.
Yes, no, thanks for this question. And it's not easy. What is early enough if you do not oversee the horizon? I remember in the very beginning, when I tried to convince my management about this project, I said, oh, it will take 5 years and a few resources. Now we are 10 years later, so it can go a bit wrong with your estimate of time horizon. But what is important, I would say, really from the beginning, is to involve a lot of stakeholders. Do not develop something in an ivory tower because you are convinced it's the right thing to do. You need to talk to the countries. We talk to so many experts in the endemic countries. they gave so good advice. I remember Professor N'Goran, when we talked about mini tablets, I will never forget the discussion we had counting 36 mini tablets for a child. This is not feasible. Who said this is couscous, but this is not a medical intervention. so this, it stuck in my head forever because you can have great ideas and it's not going to work. And you need to have this advice early on.
And this webinar is also, in a sense, a methodology or a method to interact with the environment. And could you reflect, why are we now talking at this moment on this phase of the project?
Sorry,
could you?
I did not get the question. Sorry, Bert.
Well, to continue on your response, I got also the moment of this whole webinar is also a way of interacting with the environment, because the team interacts and presents the clinical data and also the ADAPT program and the whole journey. Why are we talking at this moment in the whole project? What is the impetus to do it right now?
I think the trigger are results, of course. You have data, you can talk about this, but I think a continuous dialogue is also important.
Yes.
When we started 10 years ago, I think every year also WHO rightly ask, how long does it take? So you need to have this dialogue. You need to explain when you have delays. Why are there delays? There's nothing to hide. You have organizations like EDCTP and GIET. They are important partners. You need to explain what's happening. I think that's also a recipe of success, how we included all partners and also associated partners into the project. Yeah.
Thank you, Jutta. And Peter, could you reflect on this importance of early interactions and early dialogue? It was already in a number of your slides, but maybe some in-depth response on this.
Yes, thank you very much. We, of course, benefit a lot from the work which has already been done in the countries, and through the consortium work, all the clinical development has brought together a lot of the relevant stakeholders. What we were adding here is the social science component, really understand at the community level what the local population understands on the schistosomiasis, what are the priorities, what are triggers for them to get their children treated or not treated, so that we can react on that and take this into account when we design a strategy for the social mobilization, for awareness raising, for schistosomiasis and so on. So this is in a way a new component, a new aspect, which we feel is adding more complexity, but also adding a lot of additional insights, which we would not have without such a specialized work stream. And we don't have yet the results, of course, but we count a lot on that to help us coming up with the strategies which make the drug not only a clinical success, but also at the community level an accepted and perhaps even desired intervention. If we look at deworming in general, I think we have good examples that population accepts deworming, understands what deworming means for child development, and I see no reason why this should be different for pediatric schistosomiasis.
Yeah. Peter, in the chat, there was also one of the questions related to this conversation. Are there any plans also to go to endemic countries in Latin America? Are there any plans for doing that as well?
I mean, we have in the consortium a very strong partner from Brazil. So Latin America is represented in the consortium. The ADOPT program itself is active in African countries, as you have seen, but the drug is available, is even manufactured in Brazil, and we have very strong partners there. So I would expect in parallel that it's also being taken up into a similar type of program for then ultimately deployment in Brazil. Yes.
Yeah, excellent. I think that is very important. And Professor N'Goran, on this early dialogue, you got visitors very early in the project, and as you already said, you had not only serious questions, you had also laughs about, little pills, etc. But the overall message, and that was also reflected in some of your slides, is important. If you want to build trust, if you want to convince the local leaders, then you should have an early story with convincing not only data, but also a story that makes clear that the local communities will benefit. Could you reflect on your experience on that?
Yes, of course. I think that communication and agreement are the key elements of the success of these initiatives. Of course, at a national level, as I said, the National Ethics Committee had a different interpretation than the Directorate of Pharmacy about the authorization procedures. And since the regulation on which the agency was based was from 1970, but the Ethics Committee was based on a document that was drafted later on. So we started with the authorization from the Ethics Committee, because since it was the health ministry who authorized the procedure, we thought it was valid. But when there were undesired serious events, the Directorate of Pharmacy and Pharmacovigilance needed to issue their opinion when the trial was suspended. And the DPML finally said that it hadn't granted an authorization to the case that was submitted by the Ethics Committee. So we needed to discuss, and we needed to decide with the cabinet of the health ministry to reach an agreement and deliver the molecule for the trial. But at a community level, sometimes we were called by chiefs or important people in the community that asked us if we knew the team that was there in the village. And many times the leader of the community called to get information about what was going on, and to say that there was a team that was working in the environment. So to create trust among everybody, it was of the utmost importance to inform all the leaders of the activity that was being carried out, so that everybody agreed. So all the team members, the sponsors, and everybody, it was important that everybody interpreted the things in the same way. So it was necessary to discuss and converse to find a solution to all the incoherences and misunderstandings. But we had a meeting in Geneva, and we wanted to know there what was the kind of medicine that we needed to find. And we were talking about syrups or tablets and different things. And at that time, I had made a remark that Utah had launched a committee in 2012. Okay, yeah.
Because I think it's so important that we have that interaction between the different cultures. I come back to that point because it is also one of the discussion. Benjamin, there were a number of discussions about the clinical data, and I will direct them to you. There were much upon the exclusion in the trial, but there were also on the diagnostics, and there were a few on the PK and the dosing. Maybe the first, a quick one. Why was anti-malaria excluded? Maybe you could reflect on that.
Yeah, this is a great question, and I should have explained it. So they were not excluded because we had specific concerns. They were excluded because we wanted to understand the efficacy of arpraziquantel alone, yeah, and we wanted to exclude that there's influence on cure rates and also egg reduction rates, which is known for some of the antimalarials, like for example chloroquine, that this would have an indirect influence via praziquantel. So that's why we excluded them, not because of specific concerns, but to have clean data that show the efficacy of our praziquantel.
Yeah. Okay, and when it comes to the test itself, there was also another question in the chat box, but also which was it, PCR or PAT? So what was used?
Yeah. I mean, we did a standard method, yeah, for Manzoni. We did CAT, so stool smears, and then the eggs were counted microscopically. And then for schistosoma hematobium, we did urine filtration and counting of eggs. What is important is the sampling. So despite the fact that these are less sensitive methods, for example, compared to PCR, but also hard to implement PCR testing in such a trial, but we try to make it more sensitive. Yeah, so we used 3 samples in the week 3, but also the week 5 on 3 days. So basically, a cure was defined as negative samples on 3 independent samples taken on 3 days.
Yeah. And the question and that's of course a key question on the dosing. You know, we saw 50 milligram, we saw 60 milligram, and I see Utah also reflecting on that. So, you know, where are we now based on this data?
So, from my perspective, the way how we designed the trial program, that we just did dose finding only in schistosoma manzoni in the phase 2, and then had the schistosoma hematobium species first time enrolled in the phase 3 trial was a, let's say, efficient approach. Yeah. And then there are consequences to it. Yeah. So probably how this will look like then later is, I mean, we will propose a target label, and probably how this would look like is that indeed we have different dose levels for the 2 different species. And then for mixed infections, we would go with a higher dose. But this is because the safety looks good. But this is up for discussion, of course, and validation by the regulatory authorities. But that's a current view we have, and we can support by data. So, of course, it would have been very interesting to see with 50 mg per kg in schistosoma hematobium a later assessment time point on week 5. But we learned this during the trial, and we acted flexible to the preliminary results and to the readouts. So this is how this was structured. So I think this, what we currently can say is we have good data for efficacy and safety for the 50 mg per kg in schistosoma manzoni and for the 60 mg per kg in hematobium, and all the rest is speculation, but can be approached later.
Yeah. Utah, any additional comments from your side?
No, Benjamin's absolutely right. We need to stick to the data that we have, but we also know there is then a reality in the field. And I think there are many in this webinar who would be eager then to test also 50 milligram in hematobium with the caveat, or not with the caveat, but with the later time points, where I'm personally, I think it will work as well, but we do not have the data at the moment. So we go with what we have.
Yeah, yeah. And I think, Peter, it will also be important for your part of the program, that because given the differences in epidemiology of the different diseases, that will also have an impact on how that discussion will be for. But I think the data are straightforward, and I think the data will drive also part of the discussion, maybe also the conclusion. There was an interesting conversation, and you highlighted it also, that the whole part of the CMC and also collaboration with the Brazilian colleagues, and then going forward backwards also to the African colleagues, etc. Could you reflect on that? Because there are questions also in the chat on packaging, on different ways of how these partners will be funded or reimbursed for their efforts.
Yes, of course. And I have to say, today we do not have a solution. You know that many of these drugs for neglected tropical diseases are donated. And that's the only way how we could provide access to treatment. And it's still continuing, and we still need this. When we started this project 10 years ago, I really challenged also the community in saying, why is it always a donation at the end? What is the motivation for us to invest in R&D now and to donate everything? Aren't there other mechanisms? And I don't have the solution, but we are getting closer to this, because now we have a product. We have a local manufacturer of will get paid. The local manufacturer is not donating it. We know that the patient will not pay. So in between, we need to find a mechanism where procurement or subsidized procurement will take place. And this is also what is exactly happening now, to find ways, organizations who will buy this product, who will negotiate with countries what countries are willing to contribute to it and to take also accountability, and to find a way. We have an example with the Global Fund for Malaria, TB, and HIV/AIDS. We do not have a global fund for NTDs. But in my opinion, to encourage, to trigger innovation, to encourage more research in that field, we need mechanisms that are sustainable and where procurement is secured. And I think local manufacturing is one step. Having organizations that are interested to buy the product, or countries that may be in the position to buy the product, are ways we are looking at the moment. Yeah.
Well, thank you for that contribution. There was another question, and that was, I think that is for all panelists, I think relevant, given the nature of the project with so many different partners and continents and perspectives and cultural backgrounds, political backgrounds maybe as well, economic context, etc. How do we align with all these different perspectives, etc. And what are the roles of the individuals? And maybe Peter, you could start, and then we will give other speakers the opportunity to add.
Yes, this is actually, it's a great question, but the answer is probably not great. I can only say it's wonderful to work with such a diverse group of stakeholders, work together over years, and always it's an opportunity to learn from it. And I guess the secret behind for the successful collaboration is really this openness to learn from each other and to work together to achieve a goal which is, we all know, bigger than what each of us can achieve by him or herself or the individual institutions. So for me, this is the secret behind the success.
Mm-hmm.
Thank you.
Yeah. Daniel, could you reflect on this? Daniel?
Whom do you mean, Daniel, Bert?
Daniel. Okay, Benjamin, could you reflect on this?
Yes, great question. So I'm not so long in this.
That's why I'm asking you.
Yes, exactly. So, but I bring experience from collaborations between pharmaceutical companies. Yeah. And I think what the success factor here really in this collaboration is that we have a, let's say, alliance manager, you would call it, in a let's say, industry collaboration. And I think Lygature is really doing a great job here. So having very trustful relationships built to all the partners, having like investing a lot of time also in these one-on-one conversations before it comes to the like cross-functional, cross-partner meetings, to align, to spend a lot of work here. So I think this is, from my perspective, something that really is working very well here in this, specifically in this Pediatric Praziquantel Consortium. And I would love to see such a alliance management understanding and role also in industry collaborations. I think we can learn from this also as a pharmaceutical company here. Yeah.
Thank you, Benjamin. Professor N'Goran, do you have any thoughts on this and on the importance of bringing these different perspectives together?
I think, first of all, that having the opportunity to contribute in one's life to find a solution to a major problem among the international community is a privilege. And this enables one to potentialize one's efforts and to minimize any hindrance, any issue that would lower one's efforts. Of course, it's not always easy to adapt to the background. Look at the internet question. Someone in Germany or in any European country could not imagine that nowadays, in 2021, someone cannot have a good and correct internet access in a university. So sometimes some situations may lead each other to have doubts. But I think that trust that is developed throughout the program, throughout the interactions, helps us tolerate each other and find a solution to a major problem. And I think this is a relevant element in the success of such an experience, of such a journey.
Very encouraging for the future. And that's good news, because we need that good news also in these difficult times where collaboration, trust building, but also encouragement are so important. So thank you, Professor N'Goran, for that wonderful contribution. I see still a number of questions on One Health, but also to integrate the product in more prevention and more environmental efforts, and etc. There are some other questions on the enantiomer and on the racemate, and I must say we don't have the time to go on this, because I really feel that we should give the final word to Dr. Maganga from EDCTP to have the final word and also to reflect on what you have heard. but also to reflect for the future, because your organization is not only, in my view, funding research, but you are also you show leadership in future directions, the future scenarios to build capacity, science, but also products for global health. So the floor is yours, Dr. Maganga.
Thank you very much, Bert, and good day to everyone who is joining this very important meeting. Today marks a major milestone for the Pediatric Praziquantel Consortium on the journey towards the very big aspiration to reduce the global burden of schistosomiasis by addressing the medical needs of infected preschool children. As we celebrate jointly this major achievement of completion of the clinical development through phase 3 trial conducted in Côte d'Ivoire and Kenya, that brings the new pediatric medication closer to the patient in need, we are now very optimistic that the next milestones of registration and provision of access to suitable pediatric treatment for schistosomiasis in children 3 months to 6 years of age are now realistic targets in view. Moreover, through the ADOPT program, as we've heard, the consortium is already evaluating approaches to ensure wide acceptance and equitable access to its treatment for preschool-age children. This forward-looking 5-year program considers aspects ranging from technology transfers, logistics for local manufacturing and distribution of the drug to social mobilization and acceptance by the population. Indeed, delivery of oral dispersible praziquantel will be a very substantial contribution to the WHO Neglected Tropical Diseases Roadmap 2021 to 2030 and the United Nations Sustainable Development Goals, and in this case, targeting neglected populations in need. I wish to take this opportunity to applaud the Pediatric Praziquantel Consortium, which was established just over a decade ago in July 2012. This consortium has demonstrated key growth evolution characteristics that are geared towards creating local solutions with growing local leadership and supported by global connectivity to deliver a public good based on excellent science and innovation and in reasonable time. It has more partners from endemic countries coming on board in a shared leadership capacity. Now, with the completion of the phase 3 trial stage, the Pediatric Praziquantel Consortium demonstrates that balanced North-South collaboration with complementary expertise, bi-directional knowledge sharing, and mutual trust are key success factors to develop and deliver safe and affordable treatments for neglected tropical diseases such as schistosomiasis. On behalf of the European and Developing Countries Clinical Trials Partnership, I wish to once again congratulate the consortium for this important achievement and to restate that together with our partners at Merck, the consortium partners, the Bill and Melinda Gates Foundation, the Global Health Innovative Technology Fund, and WHO, we at EDCTP are very proud to be co-investors in the late-stage development, program that has been very fruitful. There is no doubt we felt it important to support the implementation of the Access program. The successful development and access to suitable pediatric treatment of schistosomiasis through this global public-private partnership will be a palpable contribution towards the Sustainable Development Goals by ensuring that preschool-age children, the preschool-age children group is not left behind. And with these few words, I want to thank you and hand over to Bert. Thank you, Chair.
Thank you, Dr. Makanga, and thank you, Micah, for these very important words and also very encouraging for the future. I think I mentioned also the adopt and the other aspects of the program. So thank you for that very relevant contribution. So we come to the end. I would like to thank the audience. I would like to thank the panelists, but also we had more than 100 participants all over this whole webinar. I understand the webinar becomes also available for future hearing and listening. And of course, I would also like to thank the organizers for building this wonderful program. So having said that, I close it in a formal sense. Thank you.